me here from across the hill. So I work with Nick, and most of what I present today is joint work. So I'm basically interested in understanding the evolutionary dynamics that result from selection on a very large number of loci. And one of the reasons there has been a renewed interest in this question is because of findings from very large genome-wide association studies that a lot of traits are really a lot more polygenic than people thought. So for something like human height, which is one of the best studied traits in genome-wide association studies, there is this observation that the variance explained by individual chromosomes is proportional to the length of the chromosome. So this sort of suggests a highly polygenic architecture where a lot of the variants that contribute to trait variation have small effects, and where these are sort of more or less uniformly distributed across the genome. And in fact, there are estimates that a few 100,000 common SNPs may have causal effects on height, but interestingly, very few of these actually can be detected in even very large jivas because they may fall below the threshold of significance. And so this is sort of one sort of picture that we have for some traits at least. I mean, I'm cherry picking a little bit because height is really one of the most polygenic traits. We also, increasingly, there's also a lot of interest and as well as power to look at the genetic basis of adaptation when there is selection on all this quantitative trait variation. And I guess a lot of people here also work on evolve and resequence studies where populations are experimentally evolved under controlled laboratory conditions. There are often replicates, and where the interest is often in understanding the genetic changes that underlie short-term response to selection. And these often show a sort of somewhat rather different picture where signatures of strong selection are confined to or clustered in certain genomic regions, typically tens of genomic regions. So for example, in this, sorry, what did I do?
Do you want to point?
Yeah, I wanted to.
This is old-fashioned.
Okay, sorry. Yes. So, so for example, in this plot, which I've borrowed from Kelly and Hughes, who do evolve and resequence on Drosophila similans, the sort of targets of selection which they identify are marked by these yellow triangles, and as you can see, there are, so I've sort of cut out the X because of interest of space. So as you see, there are a few targets of selection which are identified, I think, in 8 generations of selection along the genome. And this is sort of something that's seen in a variety of evolve and resequence. And one sort of question is whether this kind of heterogeneous clustered response is consistent with the picture that we had in the previous slide, where there are a lot of small effect variants that contribute to many traits. And one sort of answer that we often hear is that, well, there may be a lot of variants that are infinitesimal or basically have very tiny effects, but we also know that there are at least some major effect variants, which we might have identified with QTL mapping or something, and maybe it's these major effect variants that dominate the short-term response to selection. Of course, this raises a bunch of other questions. So one question being that, how much of the response is due to these detectable loci that we can that dominate, that we can sort of pick out using standard tests of selection, and how much of the response is due to undetectable stuff? Because if most of the response is due to the undetectable stuff, which is hard to distinguish from neutral evolution, then maybe this undetectable stuff or the background stuff is more interesting. A second kind of question is that if we look closely at any one of these peaks, then what is the finer structure here? So is it due to selection on one very atypical major effect variant, or is it due to selection on a bunch of tightly clustered, very sort of typical or modest effect variants? So that's another sort of question. And what I'll present in this talk, so the theoretical modeling I'll present is motivated by some of these questions, even though I will not necessarily answer them. So what I'm going to do in the following is to basically sort of show you that it's very useful to think of highly polygenic architectures in terms of the infinitesimal model with linkage. So this is the standard so-called infinitesimal model which I'll introduce, but we now extend it to account for the fact that because there may be a lot of causal variants that affect any particular trait, these are also not inherited independently of each other. I will then sort of look concretely at two model scenarios, one involving response from standing variation at multiple linked loci, and sort of try and figure out whether it's possible to detect selection in this limit where most of the variance comes from very small effect variants. And this will sort of naturally lead us to think in terms of blocks of the genome, and then for the rest I will focus on a single block of genome as it spreads through a genetically homogeneous population. This is, of course, a slightly artificial scenario, but it allows us to pin down what's happening in a more precise quantitative way. So I will try and sort of conclude with this. This is just a little bit about the infinitesimal model. So the infinitesimal model was first introduced by Fisher in 1918 in his foundational paper, and it has been elaborated over the next 100 years. What it says simply is that if we have a trait that is determined by a large number of loci, then there is a relatively simple description of inheritance of this trait within families. So basically, if we have two individuals, let's again take height as our example. So if we have two individuals, the genetic and whose genetic value for height is GM and GF, and if we look at their offspring and measure the trait values, that is the heights of these offspring, then the genetic component, then these will be sort of normally distributed about the parent's average, and with a certain variance, which is the within family variance, which is sort of independent of which family we are looking at, whether we are looking at a family of tall individuals or short individuals, which is more or less independent of that. And this rather simple description of inheritance of trait values emerges naturally if we consider underlying genetic architectures where there are a large number of unlinked loci of small effects, and it sort of holds even if there is selection in the population which causes the distribution of height across the population to maybe deviate from the normal expectation, there is still this simple description for inheritance within families. Of course, this assumes that loci are unlinked, and as we saw in our first slide with height, for example, that there are really a lot of variants that might affect traits, and so this assumption becomes a bit unrealistic. There may be multiple variants per chromosome. So a natural extension really is to think about the infinitesimal model with linkage. So basically assume that the trait is still affected by a lot of loci, but now explicitly account for the fact that these are sort of more or less uniformly spaced across the genome. I mean, that's an assumption that we make because it sort of simplifies the analysis, but as long as the distribution is not too clustered, it shouldn't matter. So more concretely, let's imagine that we have an additive trait that's influenced by L loci. That means that if we want to find the genetic value of any individual, we just sum over the effects of the different alleles that it carries at the L loci. we assume that there's some recombination rate between adjacent loci, and because it's a model, we can ignore all recombination rate variation along the genome. We can sort of say there are no hot spots or cold spots or whatever. And each polymorphic locus, on average, makes some contribution to the total additive variance. I'll denote this average contribution by V locus, but of course, some variants may make higher or lower contributions to trait variance than this average value, just because the effect sizes are slightly larger or the allele frequencies are slightly larger, and so on. And so the infinitesimal model or the infinitesimal limit is one where we imagine that the same total trait variation in the population is due to a larger and larger number of loci of weaker and weaker effects that are sort of more closely spaced along the genome. So basically we increase the number of loci, reduce the contribution per locus, reduce the recombination rate while keeping the total variation and the total map length the same. And the question that I would sort of want to ask is that when can selection not, or rather see or not see discrete loci, by which we mean that if we were to look at selection response due to on a population where the same total variation is due to 100 loci versus 1,000 loci, can we sort of distinguish between these alternatives? And before sort of getting into this using more sort of concrete model scenarios, I just want to say that thinking in terms of the approach to an infinitesimal limit is useful because, so whether it's with unlinked loci or linked loci, the idea is that beyond a certain effect size, can we come up with a description that is independent of genetic details that can be captured by just a few, gross parameters. And so for the infinitesimal model, we found that yes, there was this description which depends just on the variance within families. And actually it turns out that if we sort of take linkage into account, then there is, well, the description is, this is much less well studied, but there is a simple description, at least when you don't have selection, or actually more broadly, when there is not much LD in the population. So the description just is that if we randomly pick a sort of genome from the population, or a block of genome from the population, and we look at how trait value changes along this block. So basically we say that, let's imagine that we start from here and ask what's the trait value associated with this particular fragment, and so from 0 to x, or from so on, and we sort of move along the genome, then we can sort of represent it in this squiggly way. So the trait value associated with this bit here is this much. And what you see is that even if the effect sizes themselves are tiny, you can still have considerable variability along the genome, which depends basically only on one quantity, which is the total variance divided by the length of the genome. So what we are saying is that even when individual effect sizes are small, as long as there's a sufficient density of variance, you can still have considerable variability between different regions of the genome, how much they contribute to trait variance. And while this is an obvious point, it's sort of often missed when in verbal arguments about what it means to have infinitesimal trait architectures. There's this, we often sort of talk about it as if the genome would be some kind of uniform, undifferentiated mass. So this is sort of this sort of introductory kind of thing. And now I'll switch gears a little bit and talk of a very concrete simulation model, where I'll try to make more concrete this notion of how we approach the infinitesimal limit. So and I should say that this bit is preliminary work, so a lot of things are not really properly worked out. So let's imagine that we have a population with n haploid individuals, each of whom expresses some additive trait. So the trait is determined by the sum of effects of various loci on the genome. These effect size, so each locus can have 2 alternative alleles, at least in our model, and the effect sizes of the alternative alleles are drawn from some distribution with some mean. The loci are linked, that is, they are spaced over a chromosome of some map length. I'm only going to consider a single chromosome for simplicity. And the population is initially assumed to be, so we assume that there's basically no selection in the founding population, so it's in drift, mutation, recombination, balance. And then we switch on directional selection. So basically, individuals with high trait values are going to have more surviving offspring. And I should also say that, many of us study scenarios where you have a population which is basically in equilibrium, and then we, under some kind of stabilizing selection and towards some optimum, and then we shift the optimum and track the response of the population. And in this case as well, the initial response is essentially directional. So this model sort of tells us something a little more general than just, it's less restrictive than it looks. And I'm basically going to look at short-term response from standing variation, so I will neglect new mutations during this period for simplicity. And sort of to remind you, the idea again is to ask what happens to the total response to selection as well as the underlying genetic changes as we go to this limit where the same total trait variation is due to a larger and larger number of weaker effect loci that are sort of more closely spaced together. And I think this way of framing it is sort of insightful because often what we know is the total trait variation rather than the exact number of causal loci. I guess we also don't know the total trait variation at times, but there's more chance that we know that. So in this first plot, we see basically I'm plotting the change in trait mean after 50 generations of selection as a function of the number of loci that contribute to trait variation. In red, you see simulations where the loci are unlinked, as in the standard infinitesimal model. In green, you see simulations where the loci are spread across a genome of map length 0.8, and in black, the genome is just shorter. The population size is 250, and I'm just tracking the population after 50 to up to 50 generations of selection. So there are 3 things here, 3 points rather, that I would like to make. One is that, so in all 3 cases, we find that the change in trait mean under selection becomes independent of the exact genetic basis, so it becomes independent of the number of loci contributing to trait variation. This saturation occurs faster if loci are linked and if the genomes are shorter. And of course, as we expect, the response to selection is weaker when loci are linked because, when there's linkage, there's also interference between selected variants. There's so-called Hill-Robertson interference where sort of adaptive variants that are present in different regions of the genome in different individuals cannot be brought together as easily if there are if there's weak recombination. So this, of course, makes sense. So this is about, the change in trait mean. So it's basically a kind of phenotypic response. But what about genetic response? So what are the individual variants contributing to this change in trait mean? And could we detect them using some simple-minded test for selection, for example? So this is just a sort of proof of principle. So what I'm going to do is that I will look at allele frequency changes at individual selected variants, and I will call any allele frequency change as extreme if it is unlikely to have occurred under neutrality. So here I'm setting a somewhat arbitrary threshold. I call sort of an allele frequency change if an allele frequency change of a certain magnitude or higher would have occurred with probability less than 0.005 under neutral evolution, then I call this an extreme allele frequency change, and I call any locus that exhibits this kind of extreme allele frequency change as an outlier. So now what we would want to ask is that what fraction of selected variants can be detected as outliers, and how does this depend on the genetic architecture? So is it different if the same total trait variation is due to 100 or 1,000 loci? Of course, the effect sizes also become smaller as we go to higher numbers of loci. And second, what fraction of the response, what fraction of the change under selection is explained by such detectable outliers? So let me sort of go through this a bit carefully. So let's first take the case when loci are unlinked, so that they are inherited independently of each other. In this case, the fraction of variants that show this kind of outlier response decreases as the trait becomes more polygenic, and the proportion of the total response that is explained by these outliers also falls as we approach more and more infinitesimal architectures. And this is, of course, in line with our usual intuition about infinitesimal architectures, because we often, effect sizes are smaller and so on. But if you look at loci that are now placed over a single chromosome, then you see something rather different. So here, what we find is that the fraction of variants that are picked up actually reaches a constant value, which is much higher than what you would expect if you were just picking up stuff. What if you were just picking up stuff by chance, it would be 0.005, but it's really much higher. And the fraction of total response that's explained by such outliers is also significant when you assume loci that are sort of spread along a linear genome. I should say that I'm cheating a little bit because I'm choosing really high trait variation just to sort of make a certain point. Of course, if the trait variation or if selection is weaker, you would not detect as many outliers. But the point here is that it doesn't depend on the effect sizes of individual loci or the number of selected variants, but only on the total trait variation here. So we are reaching a limit where individual effect sizes don't matter as much. At least in the short run, in terms of short-term response to selection. So to sort of understand what's happening, why we are detecting so many variants, it's sort of useful to look at how these atypical variants are distributed along the genome. So here we see sort of the colored bars basically tell us the positions of outliers that are detected along the genome in a sort of simulation where the total trait variation is due to 16. In the second one, the total trait variation is due to 256 loci, and in this one, it's due to 4,000 loci. And of course, effect sizes decrease as we go up. And what we see, at least visually, is that in all three cases, there's significant clustering. We can look at this in a more systematic way by asking, how many windows have no outliers at all, how many windows have one outlier, and so on, and try and ask how much of this would be, what do we expect if we just threw them uniformly along the genome, and it is significantly non-uniform. So I want to sort of remind you that it's not, there isn't a systematic source of clustering in our simple model, so there isn't any recombination, there aren't any recombination hotspots or cold spots. In our founding population, there wasn't any systematic LD between positively selected variants because there was no selection at all in the founding population. And also when we chose effect sizes from the same single distribution. So it isn't, that there are some small effect size. There is some variance, but it's a single distribution, and we are choosing effect sizes independently at each locus. So there isn't any systematic clustering. Yet this sort of clustered response arises naturally, and so we would like to understand what is the source of this kind of heterogeneity or clustering. And one possibility is that it just, as selection proceeds, you have stochastic associations between certain variants that build up during evolution, and and it's these stochastic associations that are amplified by selection. And the other possibility is that there's something in the founding population itself, in spite of the fact that there wasn't any significant associations and so on, which causes this kind of clustering. And one way of sort of distinguishing between these two alternatives is to really look at replicates. And I'll just present one simple simulation here where we start with a population of 2n individuals. We then sort of evolve it until it's in mutation-drift recombination balance. We then split the population into two and switch on directional selection in both at the same time. And then we ask, we sort of look at outliers along the genome. And of course, there are lots of outliers because I have a relatively sort of non-stringent threshold. But let's look at the distribution of these outliers. So in red, you see outliers that are shared between the two replicates, and in gray, you see outliers that are private to one or the other replicate. And it turns out that there's a significant fraction of outliers that are shared between replicates. And one can look at this a little more systematically. And it turns out that if we just plot the fraction of outliers that appear in both replicates versus the number of loci contributing to trait variation, there's hardly any dependence, at least for this set of parameters. And this is also, so that here that 20% of outliers are shared roughly, and this is well above the significance threshold. So what this suggests is that at least in part, there is something in the founding population itself which contributes to this sort of clustered response.
Is polyclusters shorter when you have more loci, or?
So it depends on the total variation. The clusters are shorter when the variance per unit map length is low. So if you're either when the density of selected loci, so variance per unit map length just depends on basically something like so this is the effect variance per locus times the density of selected density of causal variance. So the point, I guess, is that it's one cannot disentangle these two things. I think that's the basic point of all of this. And yes, so it would be it's roughly proportional to this. And in fact, I'll sort of try to come to that in a simpler model where we can do more mathematically and understand, sort of pin down what's happening a little better. So in the last bit of the talk, I'll sort of switch to a simpler model, because what this suggests here is that selection is acting on chunks of the genome, and it doesn't really matter whether it's what matters is the total trait value associated with a chunk of characteristic length. The actual details don't matter, at least in the short run. So we would like to understand the dynamics of any one of these blocks of genome in a sort of more concrete way. And I'm going to do it using an even simpler model where basically we start with a genetically homogeneous population. We introduce a single block of genome which has sort of a very large number of weakly selected variants along it, and these sort of the trait value along this introduced block can be represented in this way. So there's some variability, different chunks may be associated with different trait values. And this sort of variability can be characterized by a single parameter, which is the variance per unit map length, or what is here. I'm going to consider a scenario where the total effect trait value associated with the block is the same as the total trait value sort of associated with any native block, so any block within the recipient population. But of course, as this block backcrosses with the native population and as it breaks up into smaller and smaller fragments, any one of these fragments may be slightly positive or slightly negative, so there can be response to selection if we, for example, switch on selection, directional selection as before. And as before, I want to sort of emphasize that I'm starting in a situation where I assume that there isn't any systematic clustering of positive or negative effect variants along the genome. So in a simulation, we just randomly randomly draw effect sizes from a distribution independently at each locus. And the two questions that I would like to sort of focus on, which are sort of inspired by the previous bit, is that, well, as this block breaks up into smaller and smaller fragments by backcrossing with the recipient population, and as each of this, so we expect that the effect sizes associated with the fragments should be smaller. The effect sizes associated with smaller fragments should also be weaker. And so in the long run, can we distinguish this from neutral introgression at all? Because individual variants really are neutral, or we can sort of consider models where individual variants are essentially neutral. Their selective effects would be just drowned out by drift or something. So that's one question. The other question is that in this infinitesimal limit where we have really tiny effect variants and there's no systematic clustering, what determines the success of different regions of the block? Can we sort of quantify this in a more precise way? And before doing this, it's useful to look at sort of certain snapshots of this process. And for those who are interested in the mathematics, we are able to model this quite using a branching process. And this sort of description is valid as long as the introduced block or its descendants are relatively rare in the population. So now this sort of picture looks at one snapshot of the process. So we see the state of the population at 20 generations and at 80 generations. So on the x-axis, we have the map. So the x-axis, as you go along the x-axis, you basically go along the introduced block. So that's the map position along the introduced block. On the y-axis, we have, so the y-axis basically identifies different descendants who carry at least some fragment of the block. So in this case, there are at least 36 descendants who carry some fragment of this introduced block. And each such horizontal line represents the colored bit represents the fragment of the introduced block that the descendant carries. The colors represent the trait values associated with different fragments. So the red bars represent deleterious fragments and the green bars represent beneficial fragments. And so what we see is that at 20 generations, there is most individuals carry rather large, weakly deleterious, or in some cases actually even strongly deleterious fragments. So this is at 20 generations. As the population evolves further, in this case, there are sort of more individuals who carry at least some fragment of the introduced block. The fragments are typically shorter, but now they are predominantly beneficial. So what has happened is that, as these blocks broke up further by recombination, some beneficial bits that might be embedded within this introduced block were loosened from their deleterious background, and then these are amplified by selection. We can sort of look at a different sort of replicate, different realization of this process, or a different replicate, which is initialized with a different introduced block, but with the same average variability between genomic windows. And here, in this case, it's qualitatively similar, except that this particular genome happened to have a particularly beneficial fragment embedded within it, which is sort of amplified quickly by selection, so that there are lots of individuals who now carry at least some fragment of the introduced block. And one can sort of go beyond looking at random snapshots and actually look at the dynamics of introgression in more detail. So by averaging over replicates, so here in black, you see the average number of descendants as a function of time in a situation where there's no fitness variation along the genome, along the introduced block. And in colored lines, you see dynamics when there is fitness variation, and as basically you go up as you increase the fitness variation, you see significant sort of deviation from the neutral dynamic. So basically, there's an exponentially fast increase in the number of descendants who carry at least some fragment of the introduced block. And I should also say that here, when I average over realizations, I'm only looking at those realizations where at least some fragment of the introduced block survives, because in many realizations you would lose the whole block, especially if you introduce it as a single copy. If you introduce it at a small frequency, not at a single copy, then this stochastic extinction is much less likely, and the average dynamics would sort of look much more like this than even if we sort of, stochastic extinction is just much less likely. So again, so this is basically, I already wrote down the results of my next slide, but I'll just try and indicate why we have these random clusters and what determines the scale of this clustering. So let's imagine that we have some fragment of map length y, and it has some selective value s. So many of these fragments will have positive trait values, many of them will have negative trait values, but the magnitude on average would be proportional to the square root of the map length, the square root of the length of the fragment, and this parameter, which is the fitness variance per unit map length. And this sort of emerges naturally if we add up a lot of random numbers. The sum is normally distributed with some typical value which scales as square root of the number of variables that we are adding up. So this is all fine, but what do we need for a fragment to be successful? We basically need that it should be amplified by selection faster than it is broken up by recombination. So if it's too small, it won't have a significant selective effect because it won't have enough variance. If it is too large, it will be broken up by recombination very fast. So there's a typical intermediate length scale that emerges such that the fragment is large enough to have a substantial selective effect, but small enough that it isn't broken up by recombination very fast. And if we just plug this S here, what we find is that the typical map length and growth rate is just proportional to this one, is basically set by this one quantity, which is the fitness variance per unit map length. And as before, we see that this involves both the variance of individual effects as well as the density of causal loci, and so it's really hard to disentangle these two things. We can also sort of go beyond these hand-wavy arguments and exactly work out what happens. So suppose I give you a particular block of genome. I say that this is effect size of first variant is this much, second variant this much, and sort of specify all the effect sizes. We can sort of work out mathematically what we expect the growth rate of any individual fragment, any individual variant within this large block to be. And it looks a bit ugly, so I don't actually show it here. But what this sort of exact solution tells us, at least for the short-term dynamics, is that, so it gives us, so it provides us with a heuristic for understanding how different variants respond, how different variants spread into this homogeneous population. So if we consider any one variant, here it's a part of many different blocks of genome. Each such block is amplified by selection or actually eliminated by selection at a certain rate, and the block is also broken up by recombination at a certain rate. So we have again this one quantity which is just the selective effect minus the recombination sort of map length. So if we look at any one such variant in the population, if we look at any one such variant along the genome, we can ask, maybe it's part of this block, and this block is really amplified by selection faster, then it's broken up by recombination. Of course, if we can also think that it's part of this large block, and this block is maybe broken up by recombination faster than it's amplified by selection. So we sort of identify the parent block that grows fastest, which is associated with the largest S minus Y value, and it turns out that in the medium run at least, the dynamics of this variant really depend only on this expected growth rate, which is the growth rate of the fastest growing parent fragment that contains it. And this intuition basically allows us to divide any genome into contiguous sort of linkage blocks, such that all variants within this block share a common medium-term fate. And as before, we see that this just depends, it doesn't depend on the details of this block, it just depends on the total selective effect of the block, which could be due to a modest number or a very large number of variants. And so, so all of this, we did some fancy maths, which was for very large populations or alternatively for very short-term spread when, the size of the population doesn't matter. So what we then tried to do was to understand whether this simple intuition also allows us to understand introgression into a finite-sized, reasonable-sized population. So we have something which is 4,000 strong, and because we don't want to have too much stochastic effect, we introduce 80 copies rather than a single copy of a particular block. And what we then ask is that if we looked at the introgression probability of any individual variants along this block, how well does it correlate with this expected growth rate, the growth rate of this parent fragment here? And it turns out that there's a very high correlation in this case in the medium run. And so which sort of tells us that this measure of linked selection, where we are thinking of the block as consisting of a bunch of, where we are considering the whole introduced block as consisting of a bunch of sort of closely linked subblocks, actually allows us to predict introgression probabilities quite well. Of course, if we have a sufficiently large population and if we track the dynamics over very long times, what we find is that this correlation is now not as amazing, because in the long run what happens is that if you have deleterious variants that are sort of embedded within these blocks, so these are denoted by cold colors here, they begin to be partially eliminated by sort of fine-scale recombination. But of course, this is happening over really long time scales. And this only happens in a sufficiently large population. So I will sort of try and conclude here. I think the time is fine. By saying that it's important to understand the effects of linkage when thinking about highly polygenic adaptation. I mean, the effects are not sort of, once one thinks about it, they are kind of obvious that, we should not be able to distinguish between blocks with a few versus a very large number of loci as long as the total trait value of the block is the same. But this sort of intuition is often missed in verbal arguments about what it means to go towards the infinitesimal limit or go towards very infinitesimal architectures. And so one of the main consequences of linkage is that it can result in detectable selection signatures even in the infinitesimal limit, and significantly heterogeneous selection response, because there's a basal null level of heterogeneity or clustering that happens even when there is no population-wide LD or no recombination, sort of complex recombination landscape. Of course, I tried to get to that in the last slide, but when we sort of talk about the infinitesimal limit, it's important to see that, it's not something that exists in a vacuum. It really depends on the size of the population. So of course, if one has a very large population, eventually fine-scale recombination will allow selection to see individual variants. If, these big blocks haven't fixed in the population. But in small populations, at least, it's sort of useful to think in terms of some of these ideas. And I should say that the last bit that I presented, so the bit about introgression, one can find more details in these two papers last year. Okay, thank you for your attention.